Hello everyone, my name is Indria Palupi. I'm honored to serve as first author and presenter of this study. Today I would like to present our research entitled Antimicrobial Activity of Neurovinegar Against Pathogenic Microorganisms and its Ability to Reduce Pro-Inflammatory Compound Expressions. In these presentations, I will briefly introduce the background of the study, explain the research methodology, present the main findings, and discuss the potential of nitrovinegar as a natural antimicrobial and a potential anti-inflammatory agent. Without further ado, let's begin with the background of this research. Let's begin with the background of this study. Indonesia is the one of the countries with the abundant natural research, including sugar palm or Eringa pinnata, which is widely distributed across several regions. One of the major production areas is the West Sumatra province, where the sugar palm plantation covers approximately 1,379 hectares. The sap obtained from this plant, known as a palm sap, has been widely utilized because its the rich nutritional composition. It naturally contains 10 to 15% sugars, making it an excellent substrate for fermentation. Its additional palm sap contains water, carbohydrates, present protein, along with other nutrients. Due to this composition, palm sap has a great potential to be developed into various fermented products, including needle vinegar, which is the main focus of this research. Although palm sap is a rich in nutrients, it's also undergoes natural fermentation immediately after tapping. During this process, sugars are the first converted into ethanol by yeast, producing its naturally fermented alcoholic beverage as a report by Twitter et al. 2020. However, the production of alcoholic beverage might raise concerns related to health as well religious considerations, limiting their acceptance in some communities. In a slide I write confidence. It means religions. Fortunately, alcohol is not the final product of the fermentation process. By continuing the fermentations under aerobic conditions using acetic acid bacteria, ethanol is further converted into acetic acid, resulting in vinegar. Previous studies have shown that several organic vinegars such as grape vinegar, pineapple vinegar, and apple cider vinegar contains bioactive compounds with antimicrobial and anti-inflammatory properties. However, information regarding needle vinegar is still very limited. Therefore, in this study, we investigate whether needle vinegar also possesses antimicrobial and anti-inflammatory potential. In addition, we evaluate the presence of lactic acid bacteria, which are commonly found in the fermented products, to determine their possible to contribution to the antimicrobial activity of needle vinegar. So, tools and materials in This study the method and tool and material we use PalmSat, which I take sample were taken from Negeri Andalai Baru Bukit Sungayang district. Tanah Datar Regency is the best quality of PalmSat, so that's why I choose this place. So the microbes and microbial media. After the natural, so we start with the process or procedure. After the natural alcoholic fermentation, the palm sap underwent acetic acid fermentation to produce neurovinegar. This experiment was conducted using completely recommends a factorial deset with two experimental factors. The first factor was a concentration of the acetobacter aceti starter consisting of 5%, 10%, 15 15% and 20%. The second factor was the fermentation durations or time, which include 3, 6, and 11 days. For each treatment, palm sap was mixed with approximately starter concentrations and then fermented according the design fermentation period. These experimental results allow us to evaluate the effect of both starter concentration and fermentation time, the quality and biological activity of neurovinegar. Next, we evaluate the antimicrobial activity of neurovinegar against three pathogenic microorganisms. The test organisms include Escherichia coli, Staphylococcus aureus as a representative bacteria, and Candida albicans as a representative fungal pathogens. Each microbial substance was spread in sterile Na- NaCl and adjust to a 0.5 McFarland standard to ensure a uniform cell density. And antimicrobial activity was then determined using the Dix diffusion method after incubation of for 24 to 48 hours. Two diameters of inhibition zone surrounding it. This was measured to evaluate the antimicrobial effectiveness of neurofinisher. Next, we isolated lactic acid bacteria or LAB from the fermentation sample with a vinegar sample where salinity dilute up to up to 10 the minus 6, 10 to the minus 4, and 10 to the minus 2. Dilution were spread into the man rugosa and serp agar or MRSA using the spread plate method. After incubating, the bacterial colonies were counted using the total plate count or TPC method to determine the population of lactic acid bacteria in each fermentation treatment. And an additional phytochemical screening was confirmed to identify the major class of bioactive compounds present in neurovinegar. GCMS analysis was then conducted to identify the major volatile and semi-volatile compounds responsible for biological activity. After identifying the major compounds, we further indicate that their potential anti-inflammatory activity using molecular tracking analysis and the lignin structure we first obtained with the PubChem database and optimized using Tram 3D, the docking simulator was then performed using Gold software against two inflammatory lethargic protein, namely TNF alpha and interleukin 6. Finally, the protein ligand interaction were analyzed and visual using LinkPlot Plus version 2.1. After completing all these analyses, I will now present the research finding, beginning with the quality bacterial characteristics of neurovinegar. Neurovinegar was actually product produced through the fermentation process. The acetic acid content obtained in this study was higher than report for salad vinegar, just 2% by Vidyari et al. 2024, indicating efficient acetic acid fermentation. However, the value was lower than the report for Kedondong vinegar at 20, 29.69% by Nugrahani et al. 2021, suggesting that substrate composition and fermentation conditions strongly influence acetic acid productions. And the antimicrobial activity of neurophagia, you can see the inhibition of neurophagia against Escherichia coli, Staphylococcus aureus, and Candida albicans. The highest antibacterial activity against E. coli was obtained after 12 days of fermentation with a 15% starter concentration, producing an inhibition zone of 13 millimeters, 13.4 millimeter. A similar trend was observed against Staphylococcus aureus with a maximum inhibition zone of 15 millimeters. In the contrast, the strongest antifungal activity was observed against Candida albicans, which produced an inhibition zone of 50.3 millimeters at 6 days of fermentation with 15% starter concentrations. The antifungal activity was substantially greater than antibacterial activity. Compared with apple cider vinegar, the antibacterial activity of Nero vinegar was higher than the inhibition zone report by Arias et al. 2022, which were just 7.42 millimeter against Escherichia coli and 10.36 millimeter against Staphylococcus aureus. Likewise, the antifungal activity observed in a distillery greatly exceeds the report by Aussie et al. 2021, who report the inhibition zone of only uh, 12 millimeter against Candida albicans. These findings indicate that needle vinegar possess strong antimicrobial properties, particularly against fungal pathogens. So we're looking for lactic acid bacteria too. And here relationship between lactic acid bacteria and antimicrobial activity. Correlation analysis revealed the complex interaction between lactic acid bacterial abundance with inhibition zone diameters. Negative correlation were observed against Escherichia coli and Staphylococcus aureus under refill fermentation condition. indicating that increased LEB population were not always associated with increased antibacterial activity. This phenomenon might be related to competition between LAB and accelerator SAT during metabolic productions. In contrast, a strong positive correlation was observed between LEB, LEB abundance and antifungal activity against Candida albicans. These findings suggest that LEB contributes significantly to fungal inhibition through the production of antifungal metabolites. Strom Strom 2025 report that LEB produce bioactive peptides capable of suppressing fungal growth. The strong antifungal activity observed in the present study might also be associated with the presence of octadecanoic acid and terpenoid compound identified through GCMS and phytochemical analysis. Previous studies have demonstrated that fatty acids such as hexadecanoic acid and octadecanoic acid act synergistically with a terpenoid to inhibit fungal growth by disrupting membrane integrity and cellular functions. Warsina et al. 2011. And the result from phytochemical screening results are referenced in my slide. The analysis confirmed the presence of saponin, tannins, phenolics, and terpenoids, while flavonoids and alkaloids were not detected. These metabolisms are known to process antimicrobial activity through various mechanisms. Saponin can increase membrane permeability and induce cell lysis, while tannins inhibits microbial lysis and protein synthesis. Phenolic compounds were reported to disrupt microbial cell walls and interfere with a metabolic process, whereas terpenoids can alter membrane structure and cellular functions. To combine and present these test compounds likely distribute to the entire microbial activity observed against the test pathogen. Similar findings have been reported by for apple cider vinegar, which contain phenolics, flavonoids, saponin, and alkaloid associated with antibacterial activity. Aliani et al. 2023. So the GCMS GCMS analysis identified five major bioactivity compounds in the neurovenature, including octadecanoic acid with 39.37%, acetic acid with 25.5%, and octadecanoic acid 12.10%, and octadecanoic chloride uh 11.87%, and hexadecanoic acid with 11.16%. The predominance of octadecanoic acid and acetic acid suggests that these compounds contribute substantially to the biological activities of neurovenature. Acetic acid is well known for its attributed properties, where oleic acid and linoleic acid have been associated with antimicrobial, antioxidant, and anti-inflammatory activities. Previous studies have report that octadecanoic acid and hexadecanoic acid inhibit fungal growth through disrupt the fungal cell walls and membranes. Warsina et al. 2011. Therefore, the abundance of this fatty acid might explain the strong antifungal activity observed against Candida albicans. And the next, in silico analysis of anti-inflammatory potentials to evaluate anti-inflammatory potency of nerve vinegar, molecular docking was performed using oleic acid and linoleic acid against the NF-alpha and interleukin 6. Oleic acid inhibited higher binding affinity toward TNF-alpha through hydrogen bond interface involving TYR1 51 and SER90 and hydrophobic interactions. Meanwhile, linoleic acid show more stable interaction with interleukin 6 through the hydrogen bonding. This interaction indicate a variable binding conformation capable of stabilizing of ligand protein complex collectivity. These findings suggest that oleic acid and linoleic acid might contribute to immuni- immunomodulatory potential of neurovenator by modulating key pro-inflammatory cytokines, namely TNF-alpha and interleukin 6. And And last is the conclusion of this study. Neurovenator produced from sugar palms or Eringa pinnata successfully met the quality increment and I01437119996 for food grade vinegar, including acetic acid content, pH, residual alcohol, and heavy metal contamination. The vinegar exhibited broad-spectrum antimicrobial activity against Escherichia coli, Staphylococcus aureus, and Candida albicans, with the strongest antifungal activity observed against Candida albicans and producing inhibited zone of 50.3 millimeters under 6 days and fermentations and 15% starter concentration. Phytochemical screening revealed the presence of saponin, tannins, phenolics, and terpenoids, while GCMS analysis confirmed octadecanoic acid, acetic acid, and octadecadionic acid as the major bioactive compounds. Correlation analysis indicate that lactic acid bacteria or LAB may contribute to antimicrobial activity, particularly against Candida albicans. Furthermore, molecular docking analysis demonstrate that oleic acid exhibits strong binding affinity toward TNF-alpha, whereas linoleic acid shows stable interaction with interleukin 6, suggesting potential anti-inflammatory activity. Overall, these findings demonstrate that needle vinegar possesses dual functionality as a natural antimicrobial and a potential immunomodulating agent, highlighting its potential application as a functional fermented product. So, thank you.